Hi, so this is Julia, and so I'm going to share the last session for today. So we're going to look at how when you alter genome sequence leads to altered functions. And so the first talk will be by Jacob Schreiber, and he's going to tell us about a method how to edit such that we can predict alterations in functional activity. So with that.
Hello, everyone. My name is Jacob Schreiber, and I'm a postdoc working with Anshul Kandaji at Stanford University. Today, I'm going to be talking about some work that I did previous to that, working with Yang Liu, back when we were both postdocs working with Bill Noble at the University of Washington. So genome editing technologies have gotten increasingly more powerful and precise in recent years. However, they leave open two major challenges. The first is identifying the set of edits that you would want to make in order to achieve some desired effect. For example, if you want to block the binding of a particular protein at some site in the genome, what edits do you need to make in order to prevent it from binding there? If you want to knock out a region of open chromatin, what nucleotides do you need to change in order to make a closed chromatin? Sometimes this is pretty obvious. If you want to knock out a protein, sometimes you can just remove the motif. Sometimes you want to do something more sophisticated, like prevent the binding of one cofactor, and you want to know how you need to change the motifs in order to prevent one of the proteins from binding, but allowing the other one to. The second issue is that sometimes the edits that you make have unintended consequences. I don't mean so much that with CRISPR-Cas9 that there are known off-target effects, where you intend to do one thing and you accidentally make edits all across the genome. What I mean is that even if you precisely made the edits that you intended to, sometimes they have unknown functional consequences that you didn't anticipate beforehand. For example, if you knock out the binding of one protein, maybe it was actually a pioneer factor that opened chromatin, and without that, it ends up turning the whole region of open chromatin into closed chromatin, preventing the binding of anything from happening there. These are two important challenges because as we try to make even more precise edits to the genome and study even more precise questions, we want to make sure that we're not confounding our results. So we propose an approach called Ledidi that tackles both of these challenges at the same time. Ledidi makes use of a pre-trained machine learning model in order to design edits. Essentially, imagine that you have a pre-trained machine learning model that takes in something like DNA sequence or RNA sequence or protein sequence, any type of biological sequence, really. You feed it into some sort of machine learning model and you make predictions for something. Maybe it's enhancer activity, maybe it's protein binding. It can really be anything. This is called the inference step. And so you take your sequence and you make predictions for what happens. Maybe you're trying to predict something like CTCF binding. This is a pretty CTCF is a pretty common protein to study. And let's say that the sequence that you ran through produced this functional profile that had two peaks. Now let's say that for the question that you were investigating, you really wanted to get rid of the left peak. And so your desired sequence looked like, your desired output from like a ChIP-seq experiment or something looked like this. You keep the peak on the right-hand side intact, but you remove the left-hand peak. How would you change the sequence in order to make this happen? Well, the way that Ledidi works is that it uses gradients between the specified signal that you want. Basically, it uses the specified signal and the original signal in order to calculate the gradient, pass that back through the model, and edit the underlying sequence. You can think about this in terms of normal machine learning. In normal machine learning, you hold your data constant and you use that to update the parameters of your model. What Ledidi does is that it holds the model constant and it uses that to update the data, in this case, the sequence that you're trying to edit. So Ledidi is not tied to any particular machine learning model, but it could be applied to any type of model any type of machine learning model where you can calculate the gradient. So specifically, what Ledidi optimizes is this objective, which is made up of two terms. On the left-hand side, you have the terms related to the number of edits made in the sequence. You have your original sequence X0 and your edited sequence X. And what you're trying to do is make the minimum number of edits that give you the desired output. On the right-hand side, you have your functional output or the outcome output. And what this is that it's the difference between what your predicted output from the model is and what your desired output is. And so what you're trying to do is you're trying to balance making the minimum number of edits while still achieving your desired goal. You have this parameter in the middle, lambda, that allows you to control, it basically allows you to weight these two terms. As you increase it, you say that it's more important to achieve your desired goal, even if you have to make more edits. As you decrease it, you say you just want to make a small number of edits, even if you don't fully achieve the desired goal. And so as a user, you're able to change that in order to balance these two terms. Now, one of the things that, one of the difficulties that we encountered is that using gradients in order to update a categorical or discrete sequence like this is not intuitive. You can't simply use gradients in order to do this. There have been a few approaches that try to overcome this, but what we chose to use was the Gumbel softmax reparameterization trick. What this does is that it basically treats every position along your sequence as a probability distribution over the potential outputs. At each iteration, what you do is you use these probability distributions in order to sample a discrete sequence, pass that through, calculate what the output is, and then calculate the gradients going backwards, but you use those gradients in order to update these underlying probability distributions. As this process continues, the distributions converge to areas where variability is okay and where variability is not okay. I'm not going to go too deep into exactly how that works, but essentially we use this trick in order to get around the difficulties of applying gradients to discrete sequences. So then we wanted to validate that Ledidi worked. What we did is that we paired it with Basenji, which is a model that takes in 131 kilobase of sequence and makes predictions for over 5,000 functional tracks. What we focused on CTCF, which is a good initial validation because CTCF is known to bind tightly to a known motif. And what we said is we wanted to do something like the initial example that I showed before. Essentially, we wanted to add in or delete peaks. So what we did is we started off doing some knockout experiments. We identified 53 sites along the genome that had strong motifs, and we wanted to eliminate the CTCF binding peaks at those locations. And so you can see that at each one of these locations, we achieved a fair amount of success. You have your original sequence on the x-axis, and the original signal, the predicted signal from the original sequence, and the predicted signal from the edited sequence on the y-axis. You can see that in almost all of these cases, we knock out the original sequence. We knock out the signal almost completely. So this means that our edited sequence gets rid of the binding of CTCF entirely. Then we wanted to look at a more complicated situation where we knock in binding. So we found locations where CTCF binding did not already occur, and we wanted to say, can we make it occur? This is a more complicated task because rather than trying to knock out a few nucleotides that would prevent CTCF binding, we have to introduce the entire motif before we would see binding. So a greedy approach that perturb nucleotides one at a time wouldn't really work because the CTCF motif is very long, and you wouldn't really see binding until you've made several correct motif nucleotide changes in correct succession. And so even though there were difficulties using gradients that had to be overcome with the Gumbel softmax reparameterization trick, that there were benefits like being able to slowly introduce a motif at multiple positions at a time. And you can see that basically we are able to add in, we're able to induce binding significantly in almost all of those positions. We then looked at what the precise edits that we made at the knockout, for the knockout experiments were. What we found is that almost all of the edits that we made around 3 edits on average per location. These edits were focused on the most conserved nucleotides within the motif on both the forward and the backward strand. So this really gave us confidence that Ledidi isn't just finding some sort of outlier thing that achieves the desired signal. It's really focusing on real biological signal because it's knocking out these conserved nucleotides. So then we tried changing the lambda parameter. And what we found is that what you would expect, as you increase lambda, you see an increased sequence loss. Basically, as you increase lambda, you're willing to make more edits in order to achieve your desired effect. But that we saw this interesting, we saw this kind of 2-hump thing with the sequence loss, the functional loss, which is on the right-hand side. What this corresponds to is that eventually, as you make a certain number of edits, you're just redesigning the entire sequence in order to achieve your desired effect. So you can see that even though you might be perfectly achieving your desired effect, that you need to constrain the amount of edits you're allowed to make. Otherwise, you just completely redesign the entire sequence. So the next thing we did is we tried using Ledidi in order to design cell type specific edits. Essentially, what we wanted to do is knock out GUND binding. We wanted to knock it out in human embryonic stem cells, but not in GM12878. And we were fairly successful at that. That at these locations, we had the same model, and we wanted to, for the same model, we wanted to keep GUND binding the same while eliminating it in another cell type at the same loci. And you can see that we significantly reduced signal in human embryonic stem cell while keeping it basically the same at GM12878. There are a lot of additional things that we want to look at here, including moving past just predicting functional annotation from genome sequence. And I think there's a lot of exciting work that could be done in this space. I'd like to thank my collaborator, Young, who helped me a lot getting all this code working, and my advisor at the time, Bill Noble, for supporting me. Thank you.
Yeah, that was a great talk, Jacob. And so let me see whether there are any questions. I don't see any questions yet. Is this right? Oh, here. Here's a question from Irina Kapler. Your CTCF knockouts were extremely successful. How often did you accidentally knock out an additional CTCF peak?
Yeah, so what we found was that in none of the cases that we looked at by hand, did we knock out the second CTCF peak in the location where there were multiple. We didn't do a more comprehensive analysis where for a large number of regions we saw whether or not additional peaks got perturbed. But we just generally didn't see that additional peaks were knocked out.
And so can you imagine doing like a motif combination? Like if there's an interaction between two motifs, could you do that?
Yeah, so one of the things that we were really interested in using Ledidi for was to try to disambiguate, basically try to look at regions where multiple cofactors were binding and see whether or not we could get some of them to bind but not others in order to find a sequence basis for this type of binding. Fortunately, what we found is that Ledidi is only going to be as powerful as the underlying predictive model. That if the underlying predictive model can't distinguish the sequence basis for different transcription factors, Ledidi can't design edits for it. And so the model that we were using, Basenji, seemed to have some difficulty with when we did some experiments with Mick and Max. And so this is definitely something that we think is a way that you can apply Ledidi in order to do scientific discovery, but we haven't yet been able to do.
That would be really exciting, yeah. So here's another question. So Melanie Weiland from the Stars Institute is saying, super interesting talk. When computing gradients, can Ledidi support other gradient methods such as deep lift or ISM?
So I'm not sure that I would call those other gradient methods as opposed to attributions, because they're trying to do something more specific than a gradient is a well-defined mathematic term. In principle, you can use attributions in the place of gradients. I'm not sure whether or not it would have to be something that you look at more carefully, because gradients have well-defined mathematic properties when you propagate them back and then use them in order to update the underlying probability distributions. I'm not sure whether or not attributions would have the same qualities.
Okay, so here's another question. Are there any non-intuitive proposed knockouts that you see? For example, a nucleotide that is not the strongest in a motif is preferred for mutation.
Yeah, so one of the things that we found with the CTCF knockouts was that rather than the top three nucleotides, the top three nucleotides in terms of conservation being the ones that I targeted, it was the top two and then kind of a middling one. And we didn't have a chance to more thoroughly investigate this yet. But what we anticipated was that this was due to the nonlinear nature of motifs. If you just look at a PWM, it's not really the case that like each of the nucleotides is independent. So what we imagined is that what's probably happening is that there's an interaction term there. That if you knock out like the most conserved and then this other this other middling one, that has a better effect than just knocking out the most conserved one. And you can see that in one of the plots that I showed in the slide.
Mm-hmm.
in the presentation.
All right, very cool. So we're going to move on to the next talk.